To explain the principal difference between alleles and genes, let's take as an example OCA2 gene. It's one of the major genes that determine eye color. As we know, the main principle in genetics is that genes regulate biological processes by making proteins. And in order to make protein, information in the gene, which is basically a part of DNA molecule, must be copied to a RNA molecule and carried to ribosomes where protein is produced. So we have this central dogma of molecular biology, DNA, RNA, protein. OCA2 gene located on chromosome 15. Chromosome itself is DNA molecule wrapped around histone proteins. So basically OCA2 gene is a particular region of DNA molecule. And DNA molecule is composed of nucleotides that are organized in two strands. First DNA strand called codon strand and contains nucleotides that form so-called codons. One codon consists of 3 nucleotides. And second strand called non-codon strand and contains complementary nucleotides that form anticodons. So we have a small region of OCA2 gene because the whole OCA2 gene is composed of approximately 380,000 nucleotides in one DNA strand. And to use this information in the gene to make protein, information must be carried to ribosomes where proteins are made. So DNA molecule for this purpose makes copy of that gene in form of pro-matrix RNA molecule. This process called transcription. And to make pro-matrix RNA molecule, RNA polymerase use non-coding strand of DNA molecule. And in this process, it uses complementary to this strand nucleotides, with only exception that thymine is replaced by uracil. So we've got pro-matrix RNA molecule. Let's suppose that our sequence of nucleotides located in coding region is called exon. So this sequence of nucleotides will not be removed by RNA splicing, and in form of matrix RNA molecule will be delivered to ribosomes. Ribosomes read nucleotide sequence and matrix RNA molecule in codons. Ribosomes read 3 nucleotides at once and include complementary to that codon amino acid. In this case it will be valine, arginine and glycine. And by this principle, ribosomes form protein. In our case, this protein called P protein, obviously it's just a small part of P protein because entire P protein is composed of 838 amino acids. P protein is necessary for melanin production in stroma of the iris. So normal P protein results in strong iris pigmentation. And everything will be that simple if we had just one chromosome. But humans are diploid organisms and we have one chromosome from father and one from mother. So there is second 15th chromosome with the same structure organization, so it also has OCA2 gene that encodes P protein. So we have one gene called OCA2 on 2 chromosomes. And despite the same structure organization, the nucleotide sequence that form OCA2 gene can be exactly the same on both chromosomes, or nucleotide sequence can be different, and the reason for it are mutations. Because mutations can change nucleotide sequence. The most common type of mutation called single nucleotide polymorphism. This mutation substitutes one nucleotide in DNA structure with another nucleotide. So let's assume that such mutation occurred on mother's chromosome at this codon, and it caused substitution of guanine with adenine. So in the second non-coding strand will be complementary to adenine thymine. In matrix RNA will be complementary to thymine uracil. And now, when ribosome reads this triplet, instead of arginine, they will include histidine into the protein structure. And now this results in production of slightly different P protein. Such mutations can totally block the protein function, slightly decrease, or in case of silent mutation, protein function may remain unaltered. Let's suppose that this mutation totally broke P protein function. Broken P protein is unable to provide melanin production, so mutated P protein results in weak iris pigmentation. By the way, such mutations are not necessarily occurred in parents' organism. More likely such mutations are inherited from generation to generation. In our case, the fact is, child has inherited altered OCA2 gene in mother's chromosome. But at which moment such mutation occurred is very hard to tell. It can be like 5,000 years ago, and from that point altered gene is inherited from generation to generation. So we have one OCA2 gene, let's call the gene that encodes iris pigmentation, on 2 chromosomes. The sequence of nucleotides that form this gene can be the same on both chromosomes, or it can be different. So basically we have 2 versions of this gene. Speaking by genetic terminology, we have 2 alleles of this gene. because allele is a version of the gene, and only one version, so-called allele of this gene, can be used for protein production. Now we assume that all other proteins that are responsible for eyes pigmentation function well. In case if we have two same alleles of this gene, everything is simple. If we have two unaltered by mutation alleles, they both encode normal P protein that will provide strong iris pigmentation, and this will manifest as brown eyes. If such mutation occurred in OCA2 gene on father's and mother's chromosomes, we have two affected by mutation alleles. They encode broken P protein that unable to provide proper pigmentation, and weak iris pigmentation will manifest as blue eyes. So if alleles are completely identical, there is no difference which allele will be expressed, because they both encode the same protein structure. But if we have two different versions of this gene, first allele is unaltered by mutation and the second allele is mutant, in this case everything depends on which allele will be expressed, or we can say which allele will be used for protein production. And interesting that even now we don't completely understand the exact mechanism, how organisms choose which allele will be expressed. But the logic is that in most cases, because mutant alleles encode broken proteins, organisms choose to use the initial version of this gene that provides strong iris pigmentation and manifests as brown eyes. So if we have two different alleles, in our case, the allele that will be expressed encodes normal P protein and called dominant allele, and the mutant allele that will not be used for protein production called recessive allele. So basically alleles is just a genetic term that means the versions of the same gene. Also we see that even mutations that cause substitution of just one nucleotide among 380,000 nucleotides can totally break function of practically any protein in human organism. And if in case of eye color it's a harmless mutation, many people find blue eyes attractive, such mutation can easily lead to hematologic malignancies as acute leukemia for example, and it's a completely different story.